One of the advantages of chemistry is that we can set problems to solve. In biological science, the problems are usually posed by nature. The chemists not only work on unlocking the mysteries of the natural phenomena, but also chemists has the ability to work on the non-natural materials they synthesize. I would call this approach the Yasuo Uchida method. Who is Mr. Uchida? If you go to a bookstore in Japan, you can easily find books written by the mystery novelist Mr. Uchida. I'm always impressed by the number of books he has written. How can he write so many books, one so quickly after another? The fact is that he has his own way of writing mysteries. Mystery writers usually build a story plot that serves as the blueprint for their writing. Their strategy usually is to give readers a clue at an early stage of the story and then reveal an unexpected twist at the end. Mr. Uchida does not build a plot. Instead, he first describes a mysterious murder scene and then tries to solve the murder himself. In effect, he creates the problems he attempts to solve. In chemistry, we can use the same method. For example, our research group stores about 70,000 chemical compounds in our chemical library freezers. I asked a postdoctoral researcher to take a picture of a chemical library we have, but I did not ask him to take a picture of himself. Most of the library members are non-natural synthetic compounds that are created using combinatorial chemistry. We have tested each of these compounds on human cells to find which of the compounds have distinct effects. Finding compounds that affect human cells does not mean that we always know how they work. We usually need to understand the mechanisms of action at molecular levels. In effect, we have created non-natural problems to solve. One such compound from our own laboratory is a synthetic compound we named phytostatin. This simple small molecule stops lipid production in cells. We initially discovered phytostatin in 2003 as a compound that inhibits the proliferation of cancer cells. However, we did not know how it worked in cells. Judging from this simple structure, we saw that phytostatin might exert its biological activity by interacting with multiple cellular proteins, and the students were afraid that they would not complete their thesis research if they work on phytostatin. Eventually, two brave researchers, one from Korea and the other from China, agreed to continue the study. By treating human cells with phytostatin, they conducted a comprehensive analysis of 30,000 human genes to examine which genes are activated and which genes are deactivated by phytostatin. Surprisingly, we found that phytostatin has an extremely simple pattern. This is a top list of inactivated genes. You can see these, the colored genes are all related to lipid synthesis. Many of the enzymes involved in lipid synthesis in the body were inactivated. The synthesis of lipids from glucose requires many enzymes. The phytostatin inactivated the genes of most of these enzymes. Pink-colored genes are actually controlled by a class of transcription factor called sterol regulatory element binding protein, SREBP. This analysis allowed us to pinpoint the SREBP pathway as a target of phytostatin. I'll tell you more about SREBP and phytostatin in the next video clip.